Hey everyone, welcome back to my channel. In today's class, we are going to start another new chapter, that is locomotion and reproduction. Before moving ahead with the chapter, I have a doubt. Yesterday, I was waving my hand and I was confused. Is waving hand is locomotion or movement? Or both are same? Let's understand this concept. The two unique features of the living organisms are your locomotion and reproduction. Hang on, ma'am, you did not speak about movement. We were confused about locomotion and movement. Don't worry, we'll study about it in upcoming slides. Now, let's first understand what is the meaning of locomotion. Probably this will give you an idea about movement and the locomotion. Locomotion is nothing but the voluntary movement of an organism. What is the meaning of voluntary? That means willingly. You want to do it by your own. Voluntary movement, we all have studied voluntary and involuntary movement. Voluntary means something which we want to do it. So that kind of movement we call it as voluntary movement. So what is locomotion? It is the voluntary movement of organism from one place to another. How do I remember it? Just remember locomotion, loco is there, na? Locomotion, loco is there, and in location again, lo is there, right? So whenever there is change in the location, that means you are moving from one place to another place, we call it as locomotion. Ma'am, still I'm confused. What about the movement? Hang on, we'll study about it. But before moving ahead, we are supposed to understand what is the reason behind locomotion. Why do we do it? Time pass. Are we doing it for time pass? We don't want to waste our energy for time pass, right? Then what's the reason of locomotion? We do it in search of food, as you can see how beautifully my cat has stolen the food. Then shelter. Shelter means home, mate, and to escape from the predator. In chat box, write what is the meaning of mate and mating. Now, introduction about the locomotion. In your textbook, they have given locomotion is influenced by internal stimuli as well as external stimuli. What is stimuli? Stimuli is nothing but any external factor which, by which we respond. External stimuli, stimuli is nothing but any external factor by which we respond. For example, when sun rays are coming, we will just close our eyes. So sun rays are going to be stimuli for us, right? Now, here are the examples of locomotion when you're walking, running, crawling, flying, and this climbing. My Nishita is climbing nicely on tree. All these are example of your locomotion, right? Now, in different animals, different type of locomotory structures are there. Now, just imagine we always walk. Suppose, let me draw over here. This is going to be Kanishk now. Okay, so if Kanish wants to walk, obviously Kanish is going to walk using his leg. Have you ever seen Kanish walking on his, have you ever seen Kanish walking using his hand? Are we supposed to do that? No, we don't do it. Animal does that, right? They use all the four limbs for locomotion. So every animal have different locomotory structure. For example, for us, it's gonna be leg, right? What about lion? Guess and write it in the chat box. Now, these structures are useful for food collection. Actually, they are doing locomotion for collecting the food. Now, the method of locomotion performed by animals varies with their habitat and demand. Now, just imagine there is an animal who stays in water, another animal who stays on land. Have you ever seen, now what, let's take an example of swimming pool. If we go in the swimming pool, what do? We swim or we are going to walk? Obviously we swim. Have you ever seen a person swimming on the land? No, obviously we need water, right? So the organisms which are there the organisms whose habitat are water or aquatic animals, they have their own locomotory structure. Then terrestrial animals, they have their own locomotory structures, right? For example, in your textbook, they have given protist. What is protist? Where we have studied protist are nothing but your kingdom protesta. Kingdom protesta I'm talking about. When I say kingdom protesta means which organisms I'm supposed to place? I'm supposed to place unicellular eukaryote. Unicellular eukaryote, right? Now, one example of protista given over here is your amoeba. Here you can see in the picture, amoeba does not have fixed shape and how beautifully its pseudopodia is growing. See here. Okay, so pseudopodia, amoeba use pseudopodia for the collection of food, right? And for locomotion. Now, when we talk about paramecium, again it is an example of protista, and cilia is the one which helps for a collection of the food, right? We are going to learn about it in detail in upcoming slides. Now, this one, all locomotion are movement, but all movement are not locomotion. Let's keep it on hold and understand this slide, then we'll go back. Now, when I asked you shaking my hand or waving my hand, whether it is a movement or a locomotion, what did we study? Locomotion is nothing but moving from one place to another. See this little cute penguin, how it is moving from one place to another place, right? So when it is moving from one place to another place, we call it as locomotion. But when you are shaking hand or during phototropism or during waving your hand, are you moving from one place to another place? No, we are not moving. That is just a process of moving. We are not changing the location. We are not changing the place, right? So when we are not changing the place, when there is no change in the location, we call it as movement. I hope you understood. Locomotion is nothing but when organism is moving from one place to another place, we call it as locomotion. But then what is movement? Organism is staying at one position and doing some move, for example raising hand or dancing at same particular place, so we call it as movement, right? For example, now understand this concept. This is the neat topic. Okay, now all locomotion are movement. When I say all locomotion, that means you are moving from one place to another place. When you're moving from one place to another place, obviously there is a movement in your body. So I can say all locomotion are movement. But all movement means you're not moving from one place to another place. So when you're not moving from one place to another place, how it can be locomotion? It won't be, right? It won't be. So locomotion is always a movement, but movement is not always a locomotion. I hope you understand this concept. Now, let's jump into another concept. Before moving ahead with the locomotion, we have to understand what all things, what all organelles or what all structures are useful for us for the locomotion. I have taken three examples over here, that is your vertebrates, invertebrates and microorganism. When I say vertebrates, who are vertebrates? Who have vertebral, spelling is wrong, vertebral column. I think it's again wrong, just correct it. Okay, vertebral column. Okay, now organisms who have vertebral column, we call it as vertebrates. What about invertebrates who do not have vertebral column? We call it as invertebrate. So when we talk about vertebrates, obviously when I saying they have vertebral column, that means the skeleton will be present. That's why our structure is like a human, not like a microorganisms, right? So we have skeleton as well as muscle, and these two are going to help in our locomotion, right? For example, human. What about invertebrates? Do they have skeleton? No, just now I told you vertebral column will be absent in them. Then what is present? Muscle is present. So what is used in locomotion? Muscle is used in locomotion. For example, your earthworm and leeches. Microorganism, in microorganisms, both skeleton and muscles are missing. So can I say they are not going to move? No, they have to move. They have to do locomotion. Otherwise, from where they are going to get food? Obviously, there will be the locomotion by using protoplasm. Now, what is protoplasm? We already studied. That is nucleoplasm plus cytoplasm. We call it as protoplasm. Now, one of the example of protoplasmic movement is your cytoplasmic movement or cytoplasmic streaming or cyclosis. Now, what are all these three? All three are same. Cytoplasmic movement, cytoplasmic streaming and cyclosis, all three are same. Now, you must have already seen cell, right? This is your cell, and not this color, let me use another color, and you have nucleus over here. What about the jelly-like fluid? It is your cytoplasm. What is cytoplasm? Cytoplasm is a jelly-like fluid present inside the cell. Now, these cytoplasm can move either clockwise manner or it can move anti-clockwise manner. Cyclosis means when cytoplasm move in a clockwise manner or anti-clockwise manner in order to equally distribute the material. What is cyclosis? What is cyclosis? Cyclic or, sorry, clockwise I should have written. clockwise or anti-clockwise movement of cytoplasm. Why do we do it? In order to distribute nutrients or substance equally. So whenever my cytoplasm is moving, suppose this is my cell and here I have nucleus, suppose this is my mitochondria. Now my mitochondria needs some protein which is being packaged by Golgi apparatus. Okay, so here you have endoplasmic reticulum and then you have Golgi, and this protein is produced. Now this protein has all the way go to the mitochondria. So how that will happen? By the movement of your cytoplasm. When your cytoplasm move, obviously the material, the protein which is formed will move towards the mitochondria, and there will be the entry of protein in the mitochondria, right? Now, animal movement can happen through amoeboid movement, flagellar, ciliary, or the muscular movement. Example, amoeba, paramecium, and bacteria, right? Now, let's understand and start our chapter with the locomotion in protozoa. But before understanding how the locomotion occur in protozoa, we are supposed to first understand what is protozoa. When we speak about the protozoa, I have already told you protozoa in which kingdom I am supposed to place it. I am going to place it under kingdom Protista. Kingdom Protista. In your textbook, they have written acellular. Don't get confused with this. Probably it's some spelling mistake or what. Protista or protozoa is not acellular. That means it is having cell. It is having cell. Probably it must be written like acellular, or maybe this means that it is unicellular. So do not get confused and say that ma'am, it does not have cell. No, it has cell. They are unicellular eukaryotic organism and they are versatile. That means flexible, easily able to do locomotion. They are versatile in their locomotion. Easily they are able to do it. Okay, easily they are able to do it. Free living, where will you find them? In aquatic. That means you are going to find protozoa in water, right? Some parasites or some protozoan you will see in your body fluid, that is your maybe your lymph, maybe your blood, maybe your digestive tract. Now blood you already know. About lymph you will study in class 12 standard circulatory system. So this lymph is formed from the blood plasma. Okay, blood plasma and WBCs and small size proteins are going to come out of your blood, and these are going to form lymph. Basically it will form tissue fluid, but this tissue fluid is going to enter into your lymphatic system, and then we call it as lymph. Right, one example I have given, that is Plasmodium falciparum is a parasite which is going to enter into your blood fluid, body fluid, that is example blood. It is going to cause malaria. The whole cycle we will study in upcoming classes. Okay, now locomotion in protozoa is performed by, so protozoa locomotion can be performed by 4 ways. What are the 4 ways? Number 1 is going to be using pseudopodia, that is cellular extension. Using cilia, that is short hair-like organelle or structure. Using flagella, that is whip-like organelle. And using myonems, that is contractile fibrils. Now for time being, understand fibrils is nothing but fiber-like structure, thread-like structure. Okay, like this, fiber-like structure, thread-like structure. Right, one by one we are going to discuss each and everything. Now protozoan exhibit 4 types of locomotion. Just now we studied about 4 organelles. See here, 4 organelles are there, or 4 structures are there which are useful for the locomotion. So if you'll see over here, pseudopodia, when an organism use pseudopodia for locomotion, we call it as amoeboid locomotion. What do we call it as? Amoeboid locomotion. Right, when an organism use cilia, we call it as ciliary locomotion. When it use flagella, we call it as flagellar locomotion. When it use myome, we call it as metabolic contractile gliding locomotion. Remember gliding locomotion because we will be using this, we will be studying about it in upcoming classes. Right, now let's understand one topic today, that is amoeboid movement, and we'll start with amoeba. What is amoeba? Amoeba is unicellular eukaryote, and you can very clearly see that it is a jelly-like jelly-like structure. Why I'm saying jelly-like structure? Because cytoplasm is what? Cytoplasm is jelly-like structure. It is unicellular. No, the more amount of cytoplasm is present, so it gives an appearance of jelly-like structure. It has no fixed structure and forms pseudopodia for locomotion. So if amoeba wants to move, what it is going to form? It is going to form pseudopodia, right? Now, three important parts which we are going to study about the amoeba. First one is cytoplasm. This is super important to understand about the cytoplasm. Now, we have seen that the cell is there and then we have nucleus, and all over here we have cytoplasm, right? Now, how is the cytoplasm in amoeba? Now, let's consider this is an amoeba. No, let me draw like this. Let's consider like this. This is an amoeba. So, cytoplasm is present. Now, the cytoplasm is divided into two parts. Cytoplasm is divided into two layers. Okay. Now, outer layer we call it as ectoplasm. You remember we studied about ectoderm. Ecto means outer layer. So, outer cytoplasm we call it as ectoplasm, right? What about the inner cytoplasm? Inner one we call it as, all over here it will be present. Inner one we call it as endocytoplasm. Endo means inside. Now, this inner cytoplasm is again further divided into two layers. Outer one we call it as plasma gel and inner one we call it as plasma soul. Gel means it is going to give the jelly-like appearance. Soul means it is going to be giving water-like appearance. Now, which one is easily able to move? Plasma soul or plasma gel? Obviously, it's plasma soul because it is in a liquid state, right? Now, let's understand the structure over here. So, this is the structure of amoeba. You can see the outer membrane, we call it as plasma membrane. It is going to be plasma membrane. Then we have this cream color which you can see here, that is your ectoplasm, that is your outer layer. The inner layer we have, which is divided into two. This thick one which you can see, this is the plasma gel like structure, we call it as plasma gel. Inner all we have water, so we call it as plasma soul. Now, this is the structure of one of the pseudopodia. Here you can see again, this one is your ectoplasm, and then you have endoplasm. Which one? Plasma gel. This whole plasma gel is present, and then you have whole plasma soul. This whole is your plasma soul, right? Now, they are moving in which direction? Obviously, in the direction where the food is present. So, obviously, it is going to be in the direction where we have to make pseudopodia, right? Now, the second structure is your plasma membrane. Plasma membrane is similar in each and every organism, let it be bacteria, be eukaryote or prokaryote, whatsoever it is. The only difference is going to be in the ratio of protein and lipid, right? So, plasma membrane is double layer and it is made up of protein and lipid. It also have nucleus which is in the endoplasm. Nucleus is present in the endoplasm. See over here. Here is the nucleus. Tell me where it is present. Is it present in the ectoplasm? No, it is present in the endoplasm. Here you can see now how pseudopodia is forming. Can you see something is coming from back and it is moving ahead, and this helps in the formation of pseudopodia? What is this whole phenomena? We are going to study now. But before moving ahead, you're supposed to know what is pseudopodia. Pseudo means false. We have studied in pseudocoelomate, right? Pseudo means fall and podia means foot. So, pseudopodia is nothing but your false foot. It is permanent or temporary? It is temporary. Extension of what? Extension of cytoplasm. Please do not write it is an extension of plasma membrane. Only plasma membrane extension will not give you the structure of pseudopodia. So, previously we studied pseudopodia is nothing but the cellular extension. When I say cellular extension here, properly if I have to say, I will be saying cytoplasmic extension, right? Now, these temporary structure is useful to move on the substratum. What is substratum? Substratum is nothing but any solid substance onto which my cell or organism move. Our cell or organism move. Okay, any substratum is nothing but any solid substance onto which my cell or organism move. Simple example if I want to give you, suppose there is a pond and you want to go from A side to B side. And there is no more water, you can walk it, but still dirty water is there. So what you will do, you are going to put let me see, where is my pencil? Here it is. Okay, so what I'm going to do, I'm going to put stones over here, and now GI is going to cross the road. Okay, so GI is going to cross the road. How? she is going to put steps on the stone, and by this she is going to cross the road. So this stone, this rock is going to act like a substratum, right? Substratum is any solid substance onto which cell or organism move ahead. Why they move? In the collection of food. Right? Now, pseudopodia is of four kind based on the structure of the pseudopodia. If the structure is blunt finger shape, please all of you draw this diagram. Blunt shape means I am talking like this. What is the meaning of blunt? Which is not sharp. If I draw like this, it is sharp, right? But here you can see it is not sharp. It is blunt. So we call it as lobopodia. The pseudopodia which has the structure of, which has a blunt finger-like structure, we call it as lobopodia. For example, your amoeba and ant amoeba. Filopodia, the pseudopodia who has the structure of fiber. For example, like this. Now, if we have fiber-like structure, we call it as filopodia. For example, euglypha. Then we have reticulopodia. If you remember in your botany chapter, we have studied about the reticulate venation. What is the meaning of reticulate? Means network-like structure. So here you can see an example that is of network-like structure like this. So, if you have this kind of pseudopodia, we call it as reticulopodia. For example, Elphidium. Okay, next one, exopodia or helopodia. When I say exopodia or helopodia, it is having the pseudopodia which is sun rays like. For example, like this. Like this. So, if the pseudopodia is like this, we call it as exopodia or helopodia or helopodia. Okay, example, actino 5. Now, basically, we are going to study how the pseudopodia is formed. Now, we are going to study about the formation of pseudopodia and how the movement occur. But before moving ahead, you're supposed to understand few concepts which are given in your textbook. Now, in your textbook, they have given the equation that is plasma gel is converted into plasma sol and plasma sol to plasma gel. Now, understand my point, what I'm trying to explain you. Let's understand with this diagram. This is the diagram of your one pseudopodia. Now, what did I tell you? Outer one is going to be plasma gel and inner we have plasma sol. If you'll see this arrow, actually what is happening? Plasma sol is moving ahead, right? Now, this plasma sol actually for the formation of pseudopodium, two processes will take place. What are the two processes? One is going to be your solation and other one is going to be your gelation. Ma'am, am I supposed to know which one is first, which one is second? No, both are happening simultaneously. Both are happening simultaneously. So, what is solation and what is gelation? Plasma gel when convert into plasma sol. Solation means convert into sol. Gel is converted into liquid, that is sol. How that will happen? Obviously, if I am going to add water. For example, in a glass you have curd, right? There is a curd and then you have water. You will see clear difference between them. But when you shake it, what will happen? The thickness of curd will be decreased. So from gel format, it will become sol format, that is liquid format. So conversion of plasma gel into plasma sol by adding water, this process we call it as solation. Then what is gelation, ma'am? Gelation means gel. So conversion of plasma sol into your plasma gel by removal of water, we call it as gelation. We call it as gelation. So this solation and gelation are helpful in the formation of pseudopodium. I am repeating again. Plasma gel is converted into plasma sol. Simultaneously, plasma sol is converted into plasma gel, and this conversion is going to help in the formation of pseudopodium. And by this, what will happen? Displacement of the body will happen in forward direction. Obviously, my pseudopodia will move in a direction where food is present. If food is present over here, is my amoeba is going to make pseudopodia over here? No, it won't make. It makes pseudopodia only in the direction where the food is present, right? Now let's see over here. So this is your plasma sol which is moving ahead. When it is moving ahead, it is getting converted here in plasma gel. Plasma gel move back and this plasma gel is converted again into plasma sol. These two things are simultaneously occurring, and this theory we call it as sol-gel transformation theory. That means sol to gel and gel to sol. This transformation, this conversion theory is going to help in the formation of pseudopodia, right? right? This is the most accepted theory. There are many theories, but you're supposed to learn about only sol gel transformation theory, right? Now, opinion differ on the site of contraction that pushes the pseudopodium, that I will be explaining you in upcoming slides. Now see here class, this is 2, 3 and 4. Now amoeboid movement, how that happened? Just now I told you that there will be a substratum. So just imagine this is the solid substance and then my amoeba is present over here. Amoeba is going to attach itself. Amoeba is going to attach itself on the substratum. It attaches itself on the substratum. Why attachment is happening? Because once the attachment is there, it is easily for me to form pseudopodia. Okay, my whole body won't move. Only the pseudopodia formation, only the cytoplasm will move ahead for the formation of pseudopodia, right? Now gelation, just now I told you what is gelation. Conversion of plasma sol to plasma gel will occur at which side? Anterior side. Anterior. This one is your anterior side of amoeba and this one is posterior. Posterior means back side, पीछे की side. Anterior means front side, आगे की side. Okay. Now, of course, my foot is over here, so there only my pseudopodia formation will occur. So this gelation process will occur, then solution process will occur, and both will occur simultaneously. Due to this, what will happen? There will be the contraction of plasma gel tube. So obviously conversion is happening, but here you have plasma gel tube. This plasma gel tube you are going to contract it. When you are going to contract it, so what will happen? Plasma cell will move forward. Plasma cell will move forward, right? Then see, I told you there will be the conversion of plasma cell to plasma gel. Let me show you the picture. I told you over here. One second, let me just erase it. Due to background, I'm unable to see. One second. Yes. Now see here, class. So I said that soul will move ahead and it will convert here in the plasma gel, but it needs something now to move soul ahead. What is that something? There will be the contraction in the plasma gel tube. There will be a plasma gel tube over here. This contracts. Due to this contraction, what will happen? Soul will move ahead and here it will turn into gel and gel will come back again and this process will takes place, right? So simple example, if I want to give you, take a balloon, blow it, and if you're pressing at one side, if you're contracting at one side, what will happen? Obviously the air will move towards the other side, right? So there will be the contraction of plasma gel tube where at the posterior side. Why? Because I have to move soul at the anterior side, right? Now see here, plasma soul is changed into plasma gel. Simultaneously, plasma gel is converted into plasma soul. We have already studied. So this soul to gel will happen in the anterior side. Soul to gel at anterior side and gel to soul posterior side. Posterior side, right? This plasma gel tube which is present over here, it will contract. That's why there will be the movement of plasma soul ahead, right? See here, plasma gel tube contracts to derive plasma soul forward. So what is happening? It is moving plasma soul forward so that it will convert into plasma gel, and by this there will be the formation of pseudopodium. See here. See here. So by conversion of plasma gel to plasma soul to plasma gel, then what will happen? Solution and gelation process will take place. By this there will be the formation of pseudopodium, and by this there will be the displacement of body in the forward direction, right? Now see class, I told you that the outer layer is going to be gel and inner layer is going to be sol. So if there is sol, that means it is a liquid. So if there is a liquid and it will be in contact with the plasma membrane, obviously my liquid will be easily able to cross through the plasma membrane. Then what will happen? My cell will shrink. There will be loss of water. There will be loss of water, due to which my cell will shrink and automatically it will die, right? I don't want that thing to happen. So what happened? Plasma gel sheet is present where? At the anterior side. Why? Because at the anterior side plasma soul is pushed, right? So my plasma soul should not cross it and meet my plasma membrane. So it prevents plasma soul reaching the plasma membrane. Sometimes what happens, this sheet can be broken down. So immediately, just now we studied, plasma soul will go there and form plasma gel. So automatically, if the sheet is broken down, plasma gel is continuously being made. So always there will be the covering of the plasma gel, right? And this helps in the formation of your pseudopodia. The other theory which is given in your textbook, but it is not explained, so you just have to remember the name of the theory, that is Allen's theory of front contraction or fountain zone theory. In soul gel, what we studied, there will be contraction in the posterior side, with that too your plasma gel tube is going to be contracted, so that plasma soul will move at the anterior position, right? Now, however, modern research bring the role of actin and myosin. About actin and myosin we will be studying in upcoming slide, but for time being, understand both are protein which helps in muscle contraction, right? Now, the pseudopodial or amoeboid movement is performed by amoeba, entamoeba, polystomela and actinophorm. So these are the four examples of your amoeboid movement. And the last topic is, we have seen in today's class that the protozoans are the one who is going to show amoeboid locomotion. Do we have any other cell who is going to perform amoeboid locomotion? Obviously, apart from amoeboid cell, we have macrophages and neutrophil. What are macrophages and neutrophils? What are macrophages and neutrophils? They are the type of WBCs. They are the type of WBCs which helps in phagocytosis. Cytosis means breaking of the cell. How? By engulfing. Engulf the foreign particle. If you can see here how neutrophil is forming amoeboid pseudopodia and it is going to engulf this bacteria. Same over here, if you can see how this macrophage is running behind this bacteria and then it is going to engulf the whole bacteria inside, right? So this was all about today's class. We have studied about the amoeboid locomotion. In next class, we are going to study about the flagella. I hope you understood the class. Thank you so much.